This e-report will be about the CRISPR-Cas9 system, how it works, and how it can be used in society today. What is CRISPR-Cas9 and what is it used for? CRISPR-Cas9 allows geneticists to edit parts of a genome by altering, adding, or replacing sections of the DNA sequence. This technology is relatively new and is still being tested for application to society. However, scientists believe that it will soon become the most common technique used to edit a genome, as it is the simplest and most adaptable method available. CRISPR-Cas9 can be used to treat diseases, as will be covered later in the presentation. However, another useful application of this system is that it can be used to help identify the functions of certain genes. By changing or removing a gene with this system, researchers can then observe the effects and identify which functions are carried out by the specific gene based on their observations. Previous methods of genetic manipulation. Prior to the recent discovery of the CRISPR-Cas9 system, geneticists have used other, less dependable and/or more expensive techniques in order to alter a genome. One method that has been used in the past is the use of chemicals or radiation to cause mutations. Although this is typically successful in mutating genes, this method provides no way of controlling which genes will be mutated in the genome, and mutations will therefore occur randomly. This type of random mutation is not typically useful for us. Gene targeting is another method that has been used in the past as a more specific method of introducing genes to a location in the genome. Gene targeting can remove or add whole genes or single bases to a genome. This is very valuable, much like CRISPR-Cas9, in identifying the functions of specific genes. However, the technology required to do this is very expensive. Creating mutations with this method is also very time-consuming. Other current methods of genome editing. Another popular method for editing a genome is performed using transcription activator-like effector nucleases, or TALENs for short. This method uses restriction enzymes that have been engineered to cut specific sequences of DNA in the target genome. Once these enzymes create a double-stranded break, the cell begins repair using the non-homologous end joining technique. If double-stranded DNA segments are introduced at the break, they may be incorporated as insertions into the genome through non-homologous end joining as well. A final method for genome manipulation is done with the use of zinc finger nucleases, or ZFNs. This method uses restriction enzymes as well, which are created by fusing specific zinc finger binding domains to a DNA cleavage domain. These zinc finger binding domains target specific DNA sequences, which are then cleaved and repaired using non-homologous end joining in the cell. As with transcription-like effector nucleases, any double-stranded DNA fragments introduced in the cell with these zinc finger nucleases will be incorporated as insertions into the genome. CRISPR and bacteria. CRISPR-Cas9 was developed from the similar natural editing system built into bacteria used to respond to invading pathogens. This system is used in bacteria as a sort of immune system, rather than as an editing technique, as it has been adapted to be used in other organisms. CRISPR stands for Clusters of Regularly Interspaced Short Palindromic Repeats. This term refers to specific regions of DNA with nucleotide repeats and segments called spacers. Spacers are bits of DNA that are scattered among these repeating sequences. Spacers in bacteria are taken from viruses that have previously attacked, serving as a sort of memory for the bacteria to recognize a specific virus if it attacks again. The function and identity of spacers was tested experimentally by a man named Rodolphe Barangu in 2007. This man and his colleagues worked at a food company called Danisco, and they worked together to perform an experiment to demonstrate the origin of spacers in bacteria. The team used Streptococcus thermophilus bacteria, which is commonly found in dairy bacterial cultures, as a model for their experiment. The scientists then observed that, after a virus had attacked these model bacterial cells, new spacers were incorporated into the bacteria's CRISPR region. Upon review, these new DNA sequences were identical to parts of the viral genome. These new spacers incorporated after the viral attack were then removed, and CRISPR's role in immunity was then confirmed, as removal of the new spacers altered the bacteria's resistance to this specific virus. It will be made in the correct place. If the Cas9 protein does not detect a PAM next to its target sequence, it will not make a cut. The CRISPR-Cas9 immunity system in bacteria. The CRISPR region in bacteria is transcribed into long RNA molecules called pre-CRRNAs. A gene from the host genome is also transcribed to create another small RNA called tracer RNA, short for transacting CRISPR RNA. The Cas9 protein is also transcribed in the CRISPR-Cas9 immunity system in bacteria. The The CRISPR region in bacteria is transcribed into long RNA molecules called pre-CRRNAs. A gene from the host genome is also transcribed to create another small RNA called tracer RNA, short for transacting CRISPR RNA. The Cas9 protein is also transcribed and translated from host DNA. With these products, tracer RNA complementary base pairs with repeat sequences in the pre-CRRNAs. The double-stranded RNA complexes resulting from this complementary base pairing become substrates for the endonuclease RNase 3 to cleave at these locations, producing short CRRNAs complexed with the Cas9 protein. In this diagram, you can see how the CRISPR-Cas9 system is used in bacteria to attack viral DNA. First, the CRISPR sequence is transcribed to create pre-CRRNA. While this is occurring, the Cas9 protein is also created through transcription and translation, and the tracer RNA is transcribed from the bacterial genome as well. Then, tracer RNA binds to pre-CRRNA and recruits Cas9 to create the units known as CRRNAs. Finally, you can see that the CRRNAs bind to the viral DNA, where Cas9 makes a double-stranded break and cleaves the viral DNA. CRISPR-Cas9 immunity system in bacteria. When a virus injects its DNA chromosome into the bacterial cell, a specific CRRNA, the tracer RNA, and the Cas9 protein will cleave the viral genome. The 5' end of CRRNA base pairs with target DNA in the viral chromosome, while the 3' end base pairs with tracer RNA to form the stem loop that binds Cas9 to the complex. CRRNA and tracer RNA therefore act together to bring the Cas9 protein to the target sequence in the viral genome. Cas9 then cleaves the viral chromosome at this location to prevent infection. Bits of viral DNA are kept and incorporated into the CRISPR region, as previously described, to help with recognition and destruction of the virus in a second attack. This system has been modified recently to be adapted for use in humans and other organisms. The CRISPR-Cas9 system has been adapted for genome manipulation. This is illustrated in the following diagram, but will be further explained in the next slides. Components of the genetically engineered CRISPR-Cas9 system. The CRISPR-Cas9 system that has been modified for use in manipulation of genomes consists first of an experimentally designed single-stranded RNA called sgRNA, short for single guide RNA. The 5' end of this sgRNA has a sequence complementary to the target site of the genome to be altered, and the 3' end binds to the Cas9 protein. This corresponds to the previously described CRRNA and tracer RNA in bacteria. Next, the system also contains a Cas9 polypeptide that has been altered to include amino acids that encode a nuclear localization signal. This signal allows Cas9 to be transported into the nucleus where it can act on DNA. That's for introducing genetically engineered CRISPR-Cas9 system into cells. There are many methods that will allow this CRISPR-Cas9 system to enter into a cell and begin manipulation of its genome. First, cells growing in culture can be transformed by plasmids containing genes that make sgRNA and Cas9. Cells with modified genomes can also be used to clone organisms. Lastly, sgRNA and Cas9 genes, transcripts, or complexes themselves can be injected into newly fertilized eggs so that they will grow into organisms with modified genomes. CRISPR-Cas9 system in the nucleus. Once in the nucleus, Cas9 sgRNA complexes seek out and bind to targeted genomic DNA sequences. The Cas9 protein then makes a double-stranded break in the target DNA that has been located by the sgRNA. The cell then responds by repairing this break by non-homologous end joining. This can result in small insertions or deletions of a few base pairs at the break. These mutations can thereby knock out the function of the affected gene. If DNA molecules corresponding to the DNA flanking the break are introduced at the same time as CRISPR-Cas9, non-homologous end joining may incorporate that DNA into the genome at the site of the break, resulting in a knock-in. The diagram illustrates the process of a genetically engineered sgRNA-Cas9 complex editing a genome. As you can see, the sgRNA is used to locate the target sequence, binding to it and bringing the Cas9 protein to this site as a result. The Cas9 protein then cleaves the target DNA, which, as shown, may result in a knockout or a knock-in. Applications of CRISPR-Cas9 in society. Use of CRISPR-Cas9 in society. CRISPR-Cas9 has great potential to treat many medical conditions that have some genetic component. Most applications being explored today use CRISPR-Cas9 to edit the genomes of somatic cells, correcting disease-causing gene mutations. Scientists are also currently exploring the potential use of this system to edit reproductive cells to prevent diseases from being passed on to offspring in the first place. However, the use of genomic editing in cells that will have such a strong effect on future generations is controversial and illegal in many countries due to this ethical debate. CRISPR-Cas9 has, however, already been used to treat the somatic cells of humans with exceptional, life-threatening cases. CRISPR-Cas9 application in fighting cancer. Scientists recently performed a test run to assess the safety of using CRISPR-Cas9 genome editing techniques to help a patient's immune system to attack cancer. Early results have indicated that the procedure was a success, but long-term results have yet to be determined. This research was paid for by the Parker Institute for Cancer Immunotherapy and the Community T-Munity. Researchers refrained from exposing the entire individuals to CRISPR-Cas9 in fear of the system being used to edit the wrong genes. Because of this, doctors removed T cells from the patient's blood and used CRISPR-Cas9 to delete 3 genes that may have been interfering with the cell's ability to fight cancer. Researchers then armed the T cells with what they called a warhead, which directs the cells to attack a protein targeted and found only on cancer cells. T cells were then processed to remove traces of CRISPR-Cas9 and infused back into the patients. Results of the experiment. Once the edited T cells were infused back into patients, researchers recorded a 10,000-fold to 100,000-fold increase in the amount of cells growing in the patients. This indicated success in spreading the genome edits to other cells. Results also indicated no negative side effects and no death of edited cells. Experiments are still being done to assess the success of these edited T cells in actually fighting cancer cells. However, researchers anticipate a great increase in the use of CRISPR-Cas9 treatment for leukemia by the end of the year. Problems to be addressed. Although the CRISPR-Cas9 system has already shown great success when applied to human cells, there is still one problem that geneticists are working to solve. The sgRNA that is used in this system contains a 20 base pair sequence, which is complementary to the target sequence to be edited. However, not all 20 of these bases need to match for the sgRNA to bind. Because of this, a sequence with 19, for example, of the 20 complementary bases may exist somewhere else in the genome. If this is the case, the sgRNA could potentially bind here rather than or in addition to the actual target sequence. This would cause Cas9 to make a cut at the wrong site and create a mutation in the wrong gene. Depending on which gene is accidentally mutated, effects could be detrimental for the individual. Solutions to ensure proper binding and cutting by CRISPR-Cas9. Solutions have been found to this problem, resulting in proper edits being made only at the target sites of a genome. This can be accomplished with the design of a more specific sgRNA, using knowledge of the sequences in the DNA genome of the organism and which genes they encode for. This can also be accomplished with the use of a Cas9 protein that will only cut a single strand of the target DNA. This would reduce the possibility of the cut being made in the wrong location, as 2 Cas9 proteins and 2 guide sgRNAs would have to be in the same location for the break to be made, thus double-checking the break. Future with CRISPR-Cas9. With this information, we could conclude that the CRISPR-Cas9 system shows great potential for treatment of diseases, as many illnesses can be treated with these slight changes in a person's genome. Ethical implications, however, may cause a slight issue, as changes in reproductive cells will be passed down from generation to generation, causing some to find it unethical to change the genome of a reproductive cell. Because of this, genome editing in reproductive cells is already illegal in the UK and in most other countries as well. Genome editing of somatic cells, however, is widely accepted and will most likely be most helpful in revolutionizing the treatment of diseases rather than preventing them. Experiments will most likely continue to be performed on humans with life-threatening cases of diseases to assess the safety and success of this type of treatment before it becomes commonly used.